Have you ever wondered why understanding gene transfer through bacterial conjugation is so crucial? Well, it's because it's a fundamental process that significantly contributes to the genetic diversity and evolution of bacteria. It's the bacterial world's version of sharing information, and it's absolutely fascinating. Bacterial conjugation, in a nutshell, is the transfer of genetic material between two bacterial cells in direct contact. It's a complex process that involves several steps, each as intriguing as the next. Firstly, the process begins with a donor bacterium that contains a special circular piece of DNA known as a plasmid. This plasmid, specifically the F plasmid or fertility plasmid, is the star of our show, as it contains the genes needed for conjugation. Once the donor bacterium comes into contact with a recipient bacterium, the F plasmid springs into action. It begins to replicate its DNA, a process known as rolling circle replication. This results in a single-stranded copy of the F plasmid, while the original remains double-stranded within the donor bacterium. Next, a structure called the pilus, which can be thought of as a microscopic bridge, forms between the donor and recipient bacterium. The single-stranded copy of the F plasmid then travels across this bridge, moving from the donor to the recipient. Once inside the recipient bacterium, the single-stranded F plasmid DNA is converted into a double-stranded DNA molecule. This transformation is crucial as it allows the recipient bacterium to become a potential donor, capable of transferring the F plasmid to other bacteria. And just like that, the process of bacterial conjugation is complete. It's a fascinating dance of DNA that happens on a microscopic scale, yet it has massive implications. To summarize, bacterial conjugation is a process where a donor bacterium transfers a copy of its F plasmid to a recipient bacterium. The F plasmid then replicates within the recipient, turning it into a potential donor. This process is one of the key ways through which bacteria exchange genetic material, contributing to their genetic diversity and evolution. Understanding bacterial conjugation is fundamental to the field of genetics. It helps us comprehend how bacteria evolve and adapt to their environments, how they develop resistance to antibiotics, and how they can be harnessed for genetic engineering. So, the next time you hear about bacterial conjugation, remember, it's not just a simple exchange. It's a complex process, a dance of DNA that plays a vital role in the life of bacteria, and by extension, in our understanding of the genetic world.